We are in an ever-changing scientific landscape where the problems that we're interested in studying are just so big. We really have to have a global society of scientists working together across different disciplines.
No single discipline can really crack the problem that we investigate. And I think about my science not through a single lens of the approaches that I'm more used to, but actually thinking more globally about problems.
The more time we spend studying these things, the more questions we can ask, the more we really begin to understand kind of the world around us and how things work. And that I find is very intrinsically exciting.
The vision of the Kavli Institute for Nanoscience Discovery is to bring the physical sciences into the cell. We can begin to understand things at the molecular level, which of course is really exciting for all sorts of therapeutics and understanding molecular processes that are going on. We have a very diverse population of scientists. So we have physicists, chemists, neuroscientists, engineers, and the goal is to get them all to come together. And we try to encourage students to drive new ideas, to influence the directions that we take.
I'm working on type 1 diabetes, which is an autoimmune disorder where the insulin-producing cells in your pancreas are destroyed. And what we're trying to do is develop these biomaterials that protect stem cell-derived insulin-producing cells, which will be transplanted to the patients and free them from lifelong insulin injections.
I'm working on point-of-care diagnostics. So that is how we can bring kind of the diagnostic technologies we have here in our hospitals and put them into a format that is low cost and easy to use for patients all over the world. And specifically, I'm working on a self-driving lab, which is basically we're removing scientists out of the equation and trying to build a robotic system to build these assays as fast and as cheap as possible.
A lot of breakthroughs are driven by the interplay between the biological question and a new technology that allows you to address it in a different way. And being in an environment like a Kavli is very important because you realize very, very quickly that nobody really cares about your scientific breakthrough. What they care about is whether your scientific breakthrough answers their question or not. And that forced me to think about, okay, how does my technology fit into this? And not just, oh, here's a shiny new piece of tech. You know, we're physical scientists developing technologies for life scientists. And normally you guess what they want, but if you're in a shared space, the barrier to just trying things out is so much lower.
So my research focuses on the molecular mechanisms of chromosome dynamics during DNA replication, and we employ interdisciplinary approaches spanning single molecule imaging, biochemistry, and molecular biophysics to understand how chromosomes are replicated. And we want to shine a new quantitative light at this most fundamental process in cellular function. So Kavli offers excellent state-of-the-art facilities which are really crucial to progress our research. Our favorite method is the microfluidics-linked turf microscopy that we combine with an in vitro frog egg extract system that enables us to conduct biochemical reactions within the microfluidic chamber and observe processes in real time. And that enables us to then address complex biological questions.
Antimicrobial resistance is becoming an increasingly large threat, and really understanding how bacteria respond to antimicrobial treatment is a really important pathway to understanding how antibiotics work and how do we design better and more effective antibiotics. So my research topic is looking at kind of stress responses in bacteria as they undergo antimicrobial pressure, particularly with regards to their cell envelope. So my project kind of bridges two techniques, so cryo-electron tomography and native mass spectrometry. So we get a lot of cellular and structural information from the cryo-ET part, and then a lot of interesting molecular insights in the native mass spectrometry part. And so bringing these two techniques together, we get a really large range of information, bridging from like big structural things to small protein level changes, yeah.
My research has a very interesting name, which is called soft landing. And so what we want to do is we want to soft land the gas phase molecule and make it as stable and as soft and as generous as possible. And at the same time, we want to keep the structure. So recently, we report the first substrate angstrom resolution membrane protein in the gas phase, and which the membrane protein can be protected in a very high resolution, and we can even achieve more because of the disordered region can be potentially protected by the polar residue in the gas phase. And the second reason is we want to combine native mass spectrometry with cryo-EM, and this kind of integration of different structure tools will make people understand more about the fundamental things of protein folding, so which I think is a revolution for the structure biology.
So we've really started to think about how the public see the science that's going on in our institute, to explore the ethics behind some of the research that we're doing. So, for example, we're working on understanding the brain.
Brain health is one of the most troubling disorders or sets of disorders to understand in modern medicine. It's so difficult to understand those processes and how they go wrong in disorders of the brain, for example, like depression or psychosis or schizophrenia. My research focuses on understanding what molecules change in the brain, particularly molecules that are involved in signaling and synapses. Our aim is to understand how these changes materialize in people with disorders, and working in the Kavli Institute provided me with connecting with individuals that are involved with psychiatry and in neuroscience, genomics, to be able to leverage molecular tools to understand disorders of the brain.
The fact that within the Kavli Institute, we are nurturing this science and society aspect where we as scientists can think about our impact beyond the bench. And it can be really difficult to consider that within discovery sciences. So to all come together and have a dedicated group thinking about science and society within the Kavli itself is a wonderful opportunity.
It stimulates this collaborative thinking and essentially enables particularly junior scientists to be brave in their scientific research. It's like a big, wonderful package of curiosity and excellence and inclusivity.
Be bold, curious, and kind. I always wanted to work in an institute that had that as its core value, and that's what we're trying to do here.